My name is Brian Guest. This is my first year here at Furman University. I have 5 undergraduates working with me this summer, 3 from Furman, 2 from the UNC Asheville. And our group is concerned with a particular type of organic compound called a cyclopropane. It's a series of 3 carbons connected in a triangular arrangement. And half of my group is working on the reactivity of those cyclopropane-containing compounds, attempting to develop new synthetic reactions that other chemists, like pharmaceutical chemists, could use to facilitate the synthesis of drugs. The other half of my group is actually using these cyclopropanes in natural products, attempting to develop anti-cancer medications that you may see on pharmacy shelves sometime in the future.
So my name is Dr. Greg Springsteen. I work in the chemistry department. My group does a variety of work, mostly related to anti-HIV medications. We do that both with organic chemistry and molecular biology. So a lot of what we work on is trying to find caps for HIV particles that prevent them from entering into human cells, and to evolve these caps, which is basically what we do. We use molecular biology techniques, and we make caps basically out of DNA, which is this informational macromolecule of the body, and we evolve these to have specific shapes. And really, we want shapes that are complementary to the surface of the HIV molecule. So we run through lots of rounds of amplifying up our DNA and mutating it and selecting it, trying to find these best molecules for binding onto this HIV so that the HIV then can enter into cells. And so for my group, there are 10 people working in my lab this summer. I have about 6 people working on molecular biology. So they're working on manipulating this DNA and selecting this DNA for the properties we want. Another 2 people are working on organic synthesis, so working on organic chemistry of molecules. They do a variety of things, including alter DNA chemically, so we can get it to do some things that it might not otherwise naturally do. We also have a couple of other projects that are related to glucose sensors for diabetes work, and so they work on making a variety of different sugar sensors. And then I have another 2 people working on automation. So what they do is they put together robotics to try to automate a lot of the things that we're doing in labs, since they tend to be very labor intensive and also extraordinarily dependent upon us not making a mess. In other words, not allowing DNA that's not part of our experiments to get into it. So if we can automate a lot of these things, we can keep them very sterile and very controlled chemical situation. So that's basically what we do. So what we have over here is just one example of what we work on. It looks like a mess, but there's actually some reason behind the madness. This is a chemical automation station. What we're doing is controlling fluids by a PC. And we just have all these pieces up to basically into solenoids, which are these pieces that allow us fluidic control. We can turn them off or on depending on what we tell the software to do, so fluid flows or not. And it's all controlled through USB into this controller card here. We also do various heating and cooling with just components we buy off the shelves, like fans and various electrical components. We work with a lot of magnets too, because we work with magnetic beads, and so we're able to stop and start again our fluidic flow and our DNA molecules by hooking them onto magnets and putting them, sending them across very, very strong 1 Tesla magnets. So we're able to get fine control of where our molecules are at any moment.
I'm Laura Wright. I'm in the chemistry department, and I'm working with students on research. And some of the students are working on projects related to the characterization of polymer films. We've been doing a lot of work with Mitsubishi polyester films, actually characterizing their surfaces using atomic force microscopy. What we're trying to accomplish is we're trying to make the samples, the films, so that they have a different surface characterization or characteristics, so that we can modify the surfaces to get better, improved properties of those samples. These polymer films are used in multiple applications. They're used in food packaging to enhance the ability of those films to prevent oxygen from getting into food samples, so they'll have a longer shelf life in the grocery stores. Also prevent water from migrating in and out of the sample, so it's something that needs to be kept so it has water in it, still has water in it. and things that don't need to have any water in them, the water stays on the outside. The films that we're working with are used for the packaging for all the Capri Sun little juice drinks, and so we're trying to make the polyester films have enhanced properties so that they can make better packaging so foods last longer, reduce spoilage.
I'm Paul Wagenknecht, chemistry professor here at Furman. We've got quite a bit of undergraduate research going on here. In my group, we specifically do photochemistry, which is really studying how light interacts with compounds. That's important really for the solar field, the solar energy field, or for photovoltaics. We study how light energy can get from one part of a molecule to another part, or from molecule to molecule. And that's really important for this whole field now of being able to collect solar energy, be able to move it around and do the kinds of things we want to if we want to try to mimic photosynthesis or to generate better photovoltaics. So a lot of the students in my group make new molecules to try to study that, and then they study what we call energy transfer between molecules and use a lot of sophisticated instrumentation. They're very good at learning these computerized instruments pretty quickly. It's impressive the things that they're able to get done, and we're making some progress on that. The National Science Foundation recently funded our group for about $270,000 over the next 3 years to work on this. So we'll have quite a few undergraduates working in this area over the next few years. It's an important problem to solve, and with enough undergraduates that learn this material and go into graduate school, we think we'll be able to make progress on it.
So I'm Jeff Petty, work here at Furman, and we're involved in research with undergraduates, mainly involved in DNA chemistry. And so one of the projects is concerned with when drugs bind with DNA, what role water plays in that process. So that's important because in a cellular environment, water is a limiting substance. The other project is also related to trying to use DNA as a template for forming new nanomaterials. Nanomaterials are impacting a lot of areas of science these days, and we're trying to use ideas from nature to try to synthesize new types of nanomaterials. And we're particularly interested in fluorescent nanomaterials, so those are the projects which we're working on. This is a high sensitivity isothermal titration calorimeter, so undergraduates use this routinely. As far as I know, we're one of the few undergraduate universities in the country that have one of these, and so we actually have two, and so you can scan over and look at the other. So we have two of these running. There are 4 groups involved in this research, and I just happen to speak among them. So.